conditions within which humanity can operate in a safe mode on the Earth. And they are all different boundaries. You can look at here, you see the climate change, you see stratospheric ozone depletion, or uh, ocean acidification. And between all these different boundaries, they all can be actually in different phases. And some of the phases are in beyond zone of uncertainty on high risk, some of uncertainty with an increasing risk, and some zones are below zone of uncertainty, safe. And if we look on the phosphorus, phosphorus is actually such critical. It's before even climate change. So there is a lot of reasons to take care on phosphorus. And what is very important once we cross the boundary, so there could be a so-called abrupt regime change, and it could be even irreversible. On the other side, if we look on the resources of phosphorus worldwide, phosphorus is non-renewable resources, is a finite resources, and even if we can find it all in all living organisms, it's actually not really common on the Earth's crust. To access all the distribution of the phosphorus, or so-called phosphate resources, is not an easy task, but we know there are more or less 11 countries which actually have the phosphate rock, and out of them, 3 actually don't export phosphorus rock. They retain phosphorus for their own use, and this is China, South Africa, and United States. So it actually leave us with 5 countries which control 90% of the world resources of phosphorus. And you can imagine that we are really heavy depend on the phosphorus. Of course, it's first of all essential for all life, and we know it can be limited, can be limiting element in aquatic environment. But our food demand, sorry, can you see that? Our food demand as actually the driving forces for the phosphorus production. And now we can see that since 1800s, the phosphorus production dramatically increasing. And if you can imagine, in the past, the human body needs 0.6, 0.7 gram phosphorus daily for our diet. Now we're at 2 to 3 grams in phosphorus. So we really heavy depend. We don't have enough resources, and they're non-renewable. What is also interesting about phosphorus, that actually phosphorus production has been always connected to social perturbation. We see here phosphorus production from 1900 till more or less present, per capita annually. And we can always connect phosphorus production and development, the social and economic development. And here the first dramatic increase is so-called green revolution. And then we got into the 70s in the stagnation around 30 kilogram per capita annually. And in 90s, last century, collapse of Soviet Union on all the East Bloc is actually led to the decrease. And now we are observing again the increase of phosphorus production, which has been connected to the development of China. But we're now in the more or less in the plateau. So talking about this, we understand how actually important, not only for our beloved science, but actually globally, to understand how is phosphorus rocks actually has been formed, and what is the global cycle. And here we are now talking about natural global cycle. If we look into the natural global cycle, we see that most of the phosphorus is actually in sedimentary rock. It's around 75%. And this rock goes through very slow weathering to the soil to be uptaken by plants and going through the food web to the animals. So those process of weathering is very, very slow, and we don't have a lot of phosphorus in the soil, and not all this phosphorus is available. Once the animals are died, we have a waste which goes through runoff to the streams, lakes, to the sediment, and once the ocean sediments are uplifting, so we can get again the sedimentary rock. There is still one, like 15%, can be created through the volcanism and this so-called indigenous rock. So, what we actually know how phosphorus has been created, and looking now into the phosphorus, into the modern sites where we know where the phosphorus is in sedimentary rock. Well, actually looking at the sediment floor in ocean. And let me find this again. And here we're looking into the areas and the locations of present-day phosphorite formation. We can see here the areas, the most of the areas are so-called upwelling area, where the nutrient-rich deep water is coming to the coast. Those areas are very important because they are creating their main sink for the global phosphorus cycle. What is interesting and exciting is that phosphogenesis or autogenic formation of phosphorus mineral is very, very complex. It has been done a lot of work to understand how actually phosphorus minerals has been formed. And of course, we talk about their phosphorus cycle, and there is a lot of phosphorus in the animals, all the bonds. So the one pretty obvious hypothesis, and actually this is also true, that all our food and our phosphorus, sedimentary phosphorus, is actually thankfully to all these nice creatures. So we can have a lot of phosphorus right now, and we can have food. So the one origin is death of marine fauna. But what is interesting, once we started looking into them more closely, into their rocks, into their phosphorites at the sea floor, we can see a lot of small grains, and the people have been interested a lot in this topic, and actually preparing our this talk and actually looking before in the history. It goes it has a long history of phosphorite, and even Vernadsky's colleague were looking into that and thinking about microbes. But one of the first work in, I hope you can see here the work of Van Capellen and Bernard showed that if we create abiotic precipitation of phosphorite, we're still getting very, very small grains. Those are results of their modeling of the experiments, where you can see here the saturation, and this is the range. We have a saturation in the sediment, in ocean sediments, poor water. And here is the size of the grains. And you can see this is a time, 500 years, 1000, 2000, but actually they're all pretty small grains, which will be created completely abiotically. But surely enough, the people looking for the microbes, and even in very, very, very old rocks, you can see here in the China, and they found a lot of fossilized bacterial cells. It leads actually to different hypotheses how it can happen, and it has been very intensively investigated also through the laboratory study and study of modern sediment. What people found, and those work has been done by Upwelling in Paris, some of this are that many bacterial cells can actually form apatite or calcium phosphate, let's say calcium phosphate. There's all the different forms. And here, for example, you can see sulfur oxidizing bacteria with a lot of granulate of so-called polyphosphate. They can be released under oxic, anoxic conditions, increasing saturation around them and pour water and actually trigger the apatite formation. It has been also in our wonderful work has been shown that actually alkaline phosphatase can control calcium phosphate precipitation. However, it was not, a lot of work has been not done in the freshwater environment. So the question about phosphorus sequestration under freshwater conditions still remains open. And in this point, since when the freshwater environment, we started to asking the question, what is actually happening in the lake, in the fresh water? What is the mechanism of phosphorus sequestration there? And one hypothesis is, which is, I think, pretty logical, since we have a lot of different observation, is that picocyanobacteria can impact the cycle and can actually serve as a phosphorus immobilization vector. First of all, they can accumulate intracellular P, and you can see here from the literature there is a different granular, and you can see that green is a polyphosphate and red is the calcium, so they are already kind of close together. And the question is, what can happen with these cells once they actually reach a sediment water interface? And we know that phosphorus can come from the polyphosphate intracellular, but calcium, maybe calcium can come from the so-called whitening events in the hard water in the lakes where we're walking on. So in order to test that, we started with the polyphosphate. And just a couple of words about the polyphosphate, because they are one of the key player in my talk. So the polyphosphate is a long chain of polymers, as you can see here. It could be 3, but it could be thousands of phosphate units. And it can be found in many cells. It could be in eukaryotes, it could be in prokaryotic cells. And it has been also found in cyanobacteria. And under TEM picture, this is some of cells in our lab. You can see them as electron-dense globulars. The PolyP accumulation can happen under two different mechanisms under culture conditions. So they could be accumulated if the cells experience very high phosphorus conditions in the media. It's so-called luxury uptake. But once the phosphorus is low in the media, the polyphosphate can be also preserved and accumulate as a granular and can use afterwards as a deficiency response. And once the cell are dying, the polyP will be released into the environment. In marine environments, there has been done research on role of polyP in the global P cycle, and it has been found that polyP is a significant component, or so-called hidden cycle of P, especially under low conditions. But there were a lot of different discrepancies and contradictory results. What is the actually reaction? Is it luxury uptake or is it deficiency mechanism? So in freshwater environments, there is not so much work has been done so far in the field. So there is a lot of questions still unclear about polyP accumulation, seasonal response, preferences of accumulation. All these questions still open in the freshwater environment. So in our approach, we first did the field study in different lakes. And here I will show you the case of Lake Ontario. In sediments to look into what is the phosphorus sinks in sediments. We also look into the surface water and we perform different lab experiments. Today, I will just show you the polyP formation driven on the P level. And I will finish with one slide about the so-called broken cycle and what we can do with all this information, trying to find a sustainable technology to actually trying to repair this broken cycle. Our field study is in Lake Ontario. We took the samples biweekly in summer. We isolated different picocyanobacteria and we extract polyP, and we also perform different lab experiments and biomineralization experiments under oxic and anoxic condition, and also in different phosphorus and calcium level to see how the external conditions actually driving internal conditions. And first of all, we found a lot of fraction of so-called calcium carbonate bound P in the sediment. It's together with organic P, it can go till 30% of total phosphorus in sediment. So it's important sink for this one of the large lake. Interesting enough, once we started doing seasonal changes, and here you see some graphs from July to December on the polyphosphate in red compared to the total phosphorus. Just pay attention, there is two different axes for polyP and for total P. They're not the same, but still it's pretty significant portion of the total P. And we also looked into the polyP in different fractions of phytoplankton, and we found that actually the small fraction of picocyanobacteria contribute most to the polyP. So it's actually very, very significant. And what is interesting, just sorry for this busy graph, don't pay attention to this part, just pay attention to this part. And also in this part, I just want you to focus on two curves. The one is this one, the black cross is um, alkaline phosphatase, and the other curve is PolyP normalized to the cell. It's actually cell PolyP, more or less. And we're looking just on the period from July to August. And what we see here in the water column in July and August, we have an increasing alkaline phosphatase. We have an increasing PolyP in the cell. If we look exactly into the mechanism, we will see this is typical phosphorus deficiency response. In blue here, this line reflects their dissolved phosphorus in water. And we can see the phosphorus goes down. This is schematical view, just to understand the process. The phosphorus, dissolved phosphorus goes down, cells starting accumulating PolyP, and alkaline phosphatase is actually increasing. And exactly during this time, and again, when we're performing the lab experiments with the cell isolated from this lake, we see exactly the same mechanisms. Once the phosphate actually goes down, the cells are starting accumulate PolyP, and alkaline phosphatase activity is increasing. Since we're getting into the conditions where we have high alkaline phosphatase, PolyP accumulation the highest, and the low P, the P stress. Under these conditions, in the lake, in the water column, we are actually observing in July 17th through the several weeks, we're observing whitening event. The whitening event, which most probable also connected to the Picocyanobacterium. And here you see this cells from the water column, this is filtered water. And what you can see, there is some morphology which is pretty similar to our Picocyanobacterial cells. They're completely covered with plates of calcium carbonate. And if you look a little bit closer, and I have like tons of all different SEM with these beautiful images, oh, I like them. So you see this small calcifying surfaces. This is like 200 nanometers, and they are all around the cells. So most probable, once they come into the sediment water interface, not so many of them will stay in the sediment. So they will be dissolving and creating calcium sources. With all this in mind, we're now going into the lab experiments and look what can actually happen with our cells in the lab. We did different laboratory experiments with our 8 strains of Picocyanobacterium. And most interesting was to push them through the conditions we know, to push them to precipitate as much, to accumulate as much intracellular PolyP as possible, and then extract that and look under different conditions, oxic, anoxic conditions, and asking the question where we can actually precipitate calcium phosphate. And with all the different experiments, you can see this, the first results through the time, the dynamic of their dissolved phosphorus in the solution. And you can see, so at one point, around 3 weeks, we see there decrease of phosphate, which it has been connected actually to precipitation of calcium phosphate. What is interesting, there's some precipitate are started already after 13 days. And these precipitate from the Picocyanobacteria strain formed actually this very small plates of calcium phosphate crystals, which are very close to the known from the literature. This is this one, which is known from the literature, which is has been formed in presence of collage, which is shows that this area has intersection with all different areas, not only um, environment and natural, but also human bodies and phosphate, calcium phosphate precipitation there. Um, we observed different precipitate under oxic and anoxic conditions, which we're still working on. Unfortunately, we didn't really uh, finish all the work we wanted to finish till now. But what was interesting, we observed different precipitates formed under oxic and anoxic conditions. Different morphologically, we don't know, unfortunately, is it really different structurally, but anyway, it's different morphologically. And we also observed, what I will not talk today, that if we're changing calcium level, we're observing so-called membrane vesicles has been formed in different Picocyanobacteria. So the last part is actually one slide I have. It's about the broken phosphorus cycle. At the beginning, we were talking about natural phosphorus cycle. Remember, we were talking about 85% of sedimentary rock, slowly weathering to the soil, plants, animal waste, and to the ocean. But what happened in with presence of humans is that we discovered that if we will add sulfuric acid to the phosphorus for rock, we can actually get a nice powder, which is can be a fertilizer. So we don't need to wait for the slowly weathering, but we can get very quick fertilizer, and a lot of phosphorus will be dissolved and will go to the soil. And with this process of fertilization and mining, we accelerate the cycle 400%. So what happens now, we have a lot of phosphorus in the soil. The phosphorus legacy in the soil will actually impact our water. You can see here the cycle now goes to the humans. We accelerate here. We're producing a lot of waste, and everything goes to the streams, lake, and to the ocean. So, and it's a long-term process, and it will be impact our water longer than the decades. And we have a so-called phosphorus dilemma. So we have a lot of phosphorus coming to the soil. The plants can take only 15%. 85% goes to the soil, through the soil to the water, creating harmful algae bloom, eutrophication, hypoxia, all the different processes which we actually don't want to have. So on one side, we have finite phosphorus resource. We don't have a lot, it's not renewable. On the other side, we are looking into our water, it's everything green. So what we can do? The one idea what we have and what we're trying to do in my lab is actually to trying to apply the process what we know to precipitate, to immobilize the phosphorus, or to so-called to trap the phosphorus. We're still at the beginning. And of course, I'm pretty optimistic that maybe we can find some technology, local technology, where we can put into the soil as a barrier, and we can defertilize the soil and trying to push our cells to create polyphosphate and actually to lease that under different conditions. And there is a hope that we can create calcium phosphate, which maybe would be useful again for the agriculture. And with this, I would like to summarize what we were talking today about. First of all, we were talking about their precipitation of calcium carbonate are close to morphology of cyanobacteria, transforming calcium to the sediment. We found a lot of calcium-bound phosphate and sediment, and representing a potential source of calcium for apatite formation. We also looked into the formation of intracellular PolyP in Picocyanobacterium, that they can actually trigger the apatite formation under laboratory conditions. And we were thinking about how to apply the mechanism, how to mimic the processes we observe in the nature to close the phosphorus cycle and trying to recover the phosphorus. And with this, I would like to thank you for your attention, and we'll be happy to have a discussion or to take any questions or comments. Thank you so much.
Thank you, Maria. Thanks for the really nice talk. If anyone has a question, either you can just raise your hand or or type it in the chat, or I can unmute you. Oh. Okay. Can everyone hear me? Okay, Maria. Okay. Well, let's see. Okay. Well, I guess everyone is thinking still. It was a lot of stuff and really good stuff. I wonder, while everyone's thinking, I just want to ask a very basic question. So you showed like how the polyphosphates in the Lake Ontario triggers the apatite formation. Is it would be a common phenomena for any freshwater environment, or is it very specific to the lake where you did the sampling?
It's actually not very specific. It's hard to generalize as always, and it's a good question. You know, I think that's a lot of processes we found as a unique, and then we find them everywhere. What we found is we found this PolyP in the lake. We did our field study on the other, and we found as well there.
Okay.
So it looks also this Picoplankton is also present everywhere. They are nice generalists, so you can find them in all the lake. You can find them also in ocean, and they're producing 50% of primary production. So it's hard to say it's a general mechanism, but what is known for long in algology that most of the cell creating PolyP.
Mhm.
Like
Yeah.
it's the mechanism is like the mechanism to have a PolyP is a general. And also like what I read recently about the so-called synthetic biology and origin of life. And the PolyP is one of the main engine. So it's one of the main energy supply for a lot of life forms.
Mhm.
And if we try to create life synthetically, like chemists trying to create life, and so the one of the sources PolyP.
Okay.
So it's it's I think it could be pretty general. It's hard to say. It's hard
Okay.
to say. It's I also didn't. It's also would be interesting to find out what is like salt or ionic strengths is doing. Like it has been done in 90s in abiotic experiments.
Okay.
Yeah.
Okay.
Mhm.
Uh,
I have a question.
Mhm.
Uh, so thank you for the presentation. It was really interesting, and I was wondering about the bioremediation you would like to apply to like erase the increase in phosphate in the soils and water. And I was wondering the phosphate due to fertilization
Mhm.
is just goes through the soil and hand in the water. So the best way to apply biomineralization to this problem would be to like use it in the soil or directly in lakes, rivers, or
Uh, it's it's a very good question, and we were thinking a lot. It's there is a lot of technology in wastewater treatment, yeah, where the people trying to get phosphorus out. It less in the agricultural areas, and the problem is that uh, the farmers they you can tell them they don't put phosphorus, yeah, you need to fertilize, yeah, so it's
yeah.
kind of um, but what is getting more and more acceptable is that in a lot of tillages, you know, like in agricultural area you have a soil and then you have a tillage where everything like goes down.
Mm-hmm.
um, in this area they are actually the main input of dissolved phosphorus as a diffuse part to the lake. So the project what we are working now with one farmer who is open to all the different ideas is actually to create kind of bioreactors from like in the area of this tillages, you know, like you have a tillages, you have this channel which bring water to the lake. So then maybe you can build some bioreactors with different biofilms or whatever, different conditions where you can increase retention time where the phosphorus will stay a little bit longer and precipitate. It has been done like, or some attempts has been done, of course, with iron,
Mm-hmm.
but then you need iron, yeah, so then you need iron. So and then you have okay, you maybe not going to vivianite, but you have some form of iron phosphate. Then it's a question, would who will apply it again? With apatite, of course it's kind of healthy fertilizer, so you could theoretically apply this again, and if farmer has it on his own, maybe it will be more motivation to reuse it.
Mm-hmm. Okay.
um, I think we have a question from Francois.
Mm-hmm.
Hi, Maria. How are you?
Hi, Francois. Good. How are you?
Very, very nice talk. Thank you very much. I had a question about also these attempts to recover the phosphates from maybe from the agricultural waste, maybe from the human waste. I don't know. Do you think that most often this would not be apatite that would form, that it would be other phosphates instead, and that have some importance then to the fate of that phosphates and maybe to their availability?
Yeah. It's a big problem. We're just starting this, and of course it will be not apatite. Yeah, as we know, it will be some octocalcium phosphate, whatever. Even in sediments, we don't have like apatite. Yeah, so in calcium P. And the question is, and the question of solubility is kind of critical. So the issue is, I think any way we can get it out of the water and kind of reuse it, like having a small internal cycle, will be helpful.
But from the literature, it's found, yeah, even there are, I heard about struvite and things like that. And so it's, but then it's interesting to, I know when you do experiments using polyphosphates as a starting material, are there special conditions to get apatite or to get other phosphates, or do you always in your own experience, for instance, do you always get the apatite, or what?
No, this is like a pain, and that's why we still didn't submit the manuscript. We did the first experiments, and then with this lockdown and ending the lab, we didn't do. But you saw from the images, we're getting all the different forms. And the issue is, that's what we are still getting, like, so we're getting all the different forms, and what is interesting and exciting, we're getting different forms based on redox conditions. And these oxic anoxic conditions, they have a different kinetic, like we're precipitating differently and different time with PolyP, and then we're getting different morphology, because our TEM is in the part of campus where they had some cases we couldn't get it, like it's we maybe next month. Morphologically, it looks completely different. It's calcium P on SEM, but it's not. I don't think it's apatite in old cases. Yeah, but this is very short time. It's kind of 13 days, 3 days.
Okay.
Yeah, but it's low calcium concentration. That what was like good news. Yeah.
Good. Okay. Thank you.
Yeah, thank you.
Do we have any other questions? Hi. Okay. And I guess we don't have any other questions, unless someone has a last minute question or something. Okay, well, with that, I think this is the time to end the seminar. Thank you so much, Maria, for a really nice talk. And have a rest of the good day. And thank you again.
Thank you. Have a good afternoon.
Thank you.
Thank you.
Okay, bye. Bye.
Bye.
Thank you. Bye.